Hello, I'm Stephen Lee, and welcome to Omics Quest. In today's Inside Ultima Genomics video, we are diving into why Ultima's cost-efficient whole genome sequencing data, currently listed at $80 per genome as of 2025, tend to have low substitution errors. Let's break it down. This video assumes you already watched another Omics Quest video titled Inside Ultima Genomics, flow-based, mostly natural sequencing by synthesis. The table shown here is taken from Ultima's WGS application note and presents benchmarking results for GIAB samples. What immediately stands out is the high precision for small indels and SNPs, which reflects effective minimization of false variant calls. It turns out that false variant calls, specifically base substitution errors, are expected to be rare in UG100 data. This is because such errors typically require adjacent homopolymer errors, which are less common in flow-based sequencing. To understand this better, let's look at how flow-based sequencing works. Each cycle consists of 4 flows, one for each nucleotide. The key challenge is determining the most likely homopolymer length in each flow. Since base calling is done by a deep neural network, it generates a probability distribution over different homopolymer lengths. Like this. From each flow's probability distribution, the system selects the length with the highest probability and assigns a conventional Phred quality score based on that probability. Now that we have covered the basics, let's explore why flow-based sequencing results in low substitution errors. Imagine we're trying to read the short sequence TAC using the UG100. It can read this in one cycle since the sequence aligns with the flow order T, A, C, and G. Since each base is read in the correct flow, the base quality scores will be high. Now, let's introduce a true biological variant, changing the final C to G. The new sequence will also be read in one cycle, maintaining high base quality. Things get more interesting if the variant changes C to T. This is still a true biological event, but to detect the second T, we must wait for the second cycle. This type of variant is known as a cyclic shift variant. In contrast, the C to G example is non-cyclic shift variant because the cycle remains unchanged. Now let's imagine these changes are not true variants, but sequencing artifacts. In the case of a non-cyclic shift artifact, where the real sequence is TAC, but the output is TAG, 2 errors must occur in sequence, missing the C and wrongly calling a G. These flows typically have low base qualities. The cyclic shift artifact is even worse. You will need to make 6 errors in a row to go from TAC to TAT. Again, effective flows will show low base qualities. Fortunately, these error patterns are easily recognized by variant calling software and can be filtered out effectively. This is why UG100 data tend to exhibit low substitution error rates. It's worth noting that in their benchmarking, Ultima used a modified version of GIAB HCR high confidence region, which excludes certain low confidence regions, specifically long homopolymers. That's because their flow-based sequencing currently struggles with reliably detecting homopolymers longer than 12 bases. Finally, in the same benchmarking, Ultima found that their CMV copy number variation calls were highly concordant with previously identified CMVs. This isn't surprising since most CMV callers rely on depth of coverage, which isn't affected by the flow-based method. In fact, higher coverage improves CMV detection, and Ultima's cost-efficient sequencing could enable even more accurate CMV analysis. Nice! Thanks for watching Omics Quest. If you enjoyed this video, please like and subscribe to the channel.